I'd like to introduce our next speaker. Dr. Jonathan Prather is an associate professor in zoology and physiology at the University of Wyoming. He received his Bachelor of Science degree in physics from the University of Virginia and his PhD in neuroscience from Emory University. He did postdoctoral work in physiology at Emory and in neurobiology at Duke University. Dr. Prather joined the University of Wyoming faculty in 2009. He teaches classes in neurobiology, as well as serving as part of the team that teaches general biology. He's the author of numerous journal articles and book chapters. He's also the principal investigator on several grants, including grants from the National Science Foundation, NASA, and Wyoming INBRE. I also learned from Dr. Prather yesterday that even though he comes from that part of the United States where there's lots of trees, he has come to enjoy the wide open spaces of Wyoming, and so we're always glad when someone comes and appreciates Wyoming. So today we're pleased to have Dr. Prather with us to discuss how the brain learns to communicate and make good decisions, and what songbirds can teach us about human behavior. John.
So thank you all for this invitation to come up and visit with you. It's my second time to Jackson, and each time that I've come, I'm struck by how absolutely inadequate cameras and words are to describe this landscape. So thank you for the joy of coming back. And today what I want to talk to you about is learning through experience. And that's something that hopefully we are pretty good at. Sometimes you learn by trial and success, and other times you learn by trial and error. And hopefully if you go through trial and error, you do so rather quickly, so you don't continue to bang your head against something. Turns out that we commonly continue to bang our heads against things, and our songbirds that we'll turn to today for understanding the neurobiology of that, they go through a staggeringly similar process. So they're more human than we realize, perhaps. So we'll begin by just talking about the context of what we're going to explore. And I think that when we are looking to look at the neural basis of learning, it's good to turn to imitative learning. And what I've shown here is this set of dancers and these people playing violin, and these behaviors are so complex that the chances of them doing the same thing at the same time just by chance is practically zero. So what we can see then is that at some point learning has occurred. At some point one of them has served as a teacher, and at another point one of them has served as a student, and they have synchronized their behavior so that they can learn to imitate one another. And that's true both for these movements and for this musical performance. So when we summarize that in neurobiology terms, we call this a sensory learning period and a sensory motor learning period. So in the sensory period, you are very passive. You are simply observing your teacher's model. It would be like having them perform the dance move while you passively watch them. And then very soon thereafter, you will attempt your own imitation of that. And as soon as you begin participating, then we enter into sensory motor learning. And you are typically rather bad at it, but then over many, many rehearsals, you get better. And that's true not only for the dance and the music that we showed, but here in our acquisition of the sounds that we use in language, this baby is positively transfixed by his father. Interestingly, when you are trying to acquire a foreign language, it is very helpful to watch the lips of the speaker. This baby somehow appears to know that. So he is cued in on his father's vocalizations coming out of his mouth, his motor performance of his lips, etc. And this baby was born able to make sound, but not able to generate anything resembling the complexity of the language that we use to communicate with one another. But the baby will eventually go through a cute phase of babbling. They'll make errors, like they'll, instead of small and little, they'll say small and whittle, things that we find very charming and very endearing. They'll go through this babbling phase, and they'll get quite good at it eventually. Turns out songbirds will do the exact same thing. Songbirds hatch, able to make sounds, but they are not able to produce the full, rich complexity of what we call song. In order to do that, a baby bird must imitate some male from around him. In many of the species around here, and all the species that we'll talk about today, males sing and females do not. So the males are the ones that are singing. Dad is typically very loud because dad is in close spatial proximity, but there are other males singing nearby, and the baby bird will form a model of this song that it would like to perform. And they're really, really good at it. Some species will learn in the late spring or early summer, and hold that in their brain for 9 months until they begin rehearsing 9 months later, at which time, without subsequent instruction, they will recall that memory, match their behavior to it so well that human ears can't tell a difference between the model and the imitation. It's staggeringly accurate, and the degree to which they can hold this in memory is quite impressive. So it turns out that Disney et al. Like to teach us that song means, oh, what a beautiful morning. And in fact, it's much more like ritualized combat, which these males are using this, yeah, I know, right? So males are using it for territorial defense, in other words, to say mine, stay away, and they're also using it to advertise their quality to eavesdropping females. We'll see later on in this discussion that females are evaluating the quality of a male song, and they're using that information to choose their mates. But for now, this is male versus male. We have a simulated territory here of male number 1 and a simulated territory of male number 2, and they are engaged in what's called counter-singing. Male 1 will say, this is my territory, and male 2 will say, okay, this is my territory, and eventually they will reach some détente status. That you're over there and I don't like you, but you're staying put, so we're cool. Interestingly, if he moves, everything else breaks loose and they have to go through it all over again. So we call this the dear enemy phenomenon. I don't like you, but stay put and we can be okay. So these males are going through this process, and if we take male 2 away, if we just go into the field and capture him for a few hours, then male 1 will sing, fail to hear his competitor sing back, and will eventually come in and take over this territory. So we see then that song is quite important in defending your territory. If we replace that male with a speaker that plays his songs, in other words, no organism is present, only the acoustic experience of song coming out of that space, then this male will respect that and not invade his territory. So from a male perspective, it's very important to learn your song and to learn it well, because this is what's going to enable you to claim territory and defend it. So then how do these males learn their songs? That's where we enter with our neuro perspective. And this slide, my wife teased me, and she said I should summarize this slide as science is complex. But the point here is that there is a section of brain, the beak would be here and the eyes would be here, and this is the cerebellum, this is the cerebellum at the back of the head, and this structure here begins at a very interesting structure called HVC. I'm just going to call HVC a series of abbreviations today. This is, if you speak human neurobiology, this is a high-level premotor cortex or associational cortex. If you don't speak neurobiology, we'll come back to it, no worries. But this gives rise to a pathway through this thing called RA, and this is in red, and eventually this is what generates the song, this red pathway. So if you get rid of any part of this red pathway, then the bird can no longer sing. He can make sounds, but he can't perform those rich, complex vocalizations that he memorized long ago that we call song. There's also another pathway from the premotor cortex down into the avian basal ganglia, down into the thalamus, back to the premotor cortex. The point being that blue eventually cycles back onto red. If you get rid of any part of the blue, then the bird can still sing, but he can't learn. That's a very interesting phenomenon. So we have these pathways, one of which is devoted primarily to the performance of the song, and the other of which, in blue, is devoted primarily to the learning and the perception and the plasticity of this behavior. So we're going to go in and we're going to record the activity of these birds as they are engaged in their avian lives. We, with our colleagues at Duke and our colleagues at MIT, we developed a very small device with electrodes finer than human hair that can go in and record from neurons as these birds are going about their lives, eating, drinking, flying short distances, singing, etc. And that's shown here. Here's a penny for scale, and this is a little device that weighs about 1 gram. It's very, very light, and we put it atop the bird's head. And the bird goes through his daily life where he's tethered, but he's free to move. We have a microphone where we record his songs if he sings. We have a speaker that we can play him songs, and we have a camera so that we can eavesdrop. Birds are jumpy critters, and if we just peered in to see what they were doing, they would stop doing what they were doing. So we put them in this private space, and they will eventually sing, and we can record the associated neurobiology. So what we see is when we record these songs, we can play them back to them, and that's shown here as a period of silence, and then this would be the voltage wobble as you recorded through a microphone, and then a period of silence. So here is one song that I'm playing, and this song is part of his adult repertoire. He's gone through juvenile experience, he's created that model, he's gone through sensory motor learning, he's a full-fledged adult, and he has a repertoire. And this one song type in his repertoire, when I play it, you see some sort of background hiss of the brain, and then pop, 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 pop. And this song sounds like tweet, 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 tweet. It's a series of syllables over and over. So each time I play a tweet, his brain responds with an action potential, a piece of very brief electrical excitation. So this is a grid of what it looks like when I play this song a lot of times. And I said the song is a series of tweets. If I show you then per tweet, per syllable down here, then we've got frequency on the y-axis and time on the x. This is about 100 milliseconds, so it's relatively brief. So he sweeps downward in frequency and then upward in frequency. Then he has a very brief note and then a longer note there. And every time I play that syllable, he responds with extreme temporal precision. So this is interesting in that this cell on the left is responding to this song type in his repertoire, which I've chosen to call the primary song type. And then on the right, I've shown you another element in his vocal repertoire. So we know he can perform this thing. We know that a neural representation of this behavior exists somewhere in the brain, but this cell represents this behavior, but not this one or any other behavior in his adult repertoire. So we have an exceedingly selective representation, very precise. This cell is representing one behavior from among a very large or a relatively large repertoire. And it's doing so with not only great selectivity, but also great temporal precision. So we have a very crisp, a very sparse representation of this behavior. And that has implications for helping you to learn, which we can talk about later if you want. But so far, we've got part of the learning process. We have auditory experience making it into the brain and the brain registering that. So what happens to these cells when the bird sings? And it turns out that this is the same cell that I just showed you. Here, I'm playing his primary song type to him, tweet, 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 and the cells going pop, 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 pop. And then he sings a series of introductory notes, and then he sings the same song type right back to me. So this is a very efficient means of asking how this cell represents one and the same behavior as either a sensory experience or a motor performance. And it turns out that this cell is representing both the sensory aspect of it and the motor encoding aspect of it. And remember, in the sensory domain, it was very selective. It would respond to one song type and not others. And when we look in the motor domain, when he sings, here, this is a different cell, different bird, but this cell is singing, the bird is singing the primary song type for this cell, or excuse me, he's hearing the primary song type, then he sings something else and the cell is off. Here he hears something else, and then he sings the primary song type and the cell turns on. So the specificity that we saw in the auditory domain is also reflected in the motor domain. And it turns out that in every single case, the song type that I called primary because of the cell's behavior when the bird heard the song was also the primary in the motor domain. In other words, these cells are representing one and the same behavior as both a sensory incoming experience and a motor outgoing performance. And again, the temporal precision that we saw in the auditory domain, very precise timing with regard to the syllable, very precise timing with regard to the next syllable and the next. The motor domain, that activity was also tightly aligned with that auditory experience. So all of that beautiful, precise activity that we saw in the auditory domain as an inbound sensory experience is also reflected in the motor performance. And so we think that there are cells that have been popularized in the human literature called mirror neurons, which is, sometimes it's called monkey see, monkey do, but it's a means of potentially interpreting experience in the outside world by your own repertoire of possible behaviors. We're exploring the idea that these may in fact be those types of neurons, and we could unpack the functional role of those cells. Another way of saying it is that these cells, HVC cells, could forge a link between perception and action. And this sounds an awful lot like learning. If you are able to perceive something and modify your behavior accordingly, it's a very elegant possibility that there is one in the same place where those two aspects are co-represented. That appears to be the case here, and we're able to explore that. But the first thing we want to ask is, before we get carried away, is this activity that we see in the auditory domain really related to perception? Not just that sounds are hitting you in the head, but that you are decoding them into something that has meaning, perception. So to do that, we're going to turn to a behavior called categorical perception. And in this case, continuous changes in a stimulus are perceived as discrete categories. So what does that mean? Let's imagine that we have some variable and we're moving it from left to right. And I'm changing the physical parameters of that stimulus by moving left to right. And then on this other axis, we'll plot what you detect, what you perceive. So if you have continuous perception, then as I move continuously, you detect a continuous difference. We have that ability. Things like if we if I were to sing, which I will not do, but if we were to begin singing, you could hear continuous changes in pitch. Categorical perception, in contrast to that, would look like as I move the physical parameters of this stimulus, you go from one percept, perceiving one thing, in a step-like fashion to perceiving another. That would be moving from one category of perception to another category of perception. And this is enormously beneficial because when we are speaking to one another, the data are literally flying past our heads, right? You get one shot at detecting what I'm saying. And if you succeed, great. And if not, it's not like text where you can read it again. The data are gone. So you would benefit from a means of taking all the variability and all the rich behaviors that we all do differently and collapsing them into something that's easy to detect. So for instance, I'm from the American South and I sound sort of like it. If I were from Boston, I would sound different. If I were from, pick your other favorite regional accent, then I would also sound different, and I could say dog or dog, and you would be able to pierce through the variability of what I just did and detect that I'd said the same word 3 different ways. That's categorical perception. It's fundamental to how we perceive the sounds we use in speech. Turns out it's fundamental also to how these birds perceive their songs. So I can illustrate this to you if I have a volunteer that would be willing to play. So just to know what you're getting into, I would like someone who has really crisp hearing to close your eyes and tell me what you hear in a series of sounds that I play. Would anyone be willing to play? You can be right there. You'll be fine. So what I'm going to do is there are numbers on this screen from 2 to 40. And what we're going to do is we, humans, make things that are parts of our speech that are called phonemes. They're the smallest elements of our vocal communication. So one of them is pa, as in potter. And the other one that we'll explore is ba, as in bottle. Each case, I'm parting my lips and p, right? And I'm starting the vibration of my vocal cords, but in one of them, pa, I'm doing it almost at the same time. In other words, there's very close to 0 time difference between when I pop my lips and when I vibrate my vocal cords. In ba, there's a much longer duration. I pop my lips, wait a while, turn on my vocal cords. It's beyond our level of intention. We don't mean to do this, but this is how we make these two sounds. So just to let you know what you're getting into, this is a synthetic version of one of those sounds.
Ba.
Wow, okay. And this is a synthetic version of the other.
Pa.
Did you hear them as different?
Ba, ba.
Ah, very good. You're ready to go. Now, what I'm going to do is please close your eyes, and I'm going to jump around the screen and play different ones, and you tell me what you hear, bottle or potter. Sound good? Yes, ma'am. All right. So here's the first one.
Ba.
B.
And the second one.
Pa.
P.
And again.
Ba.
Ba.
And
Pa.
you want it again?
That's in the middle. It's closer to pa.
All right. How about this one?
Pa.
Oh. Yes.
All right. All right. That was very. Thank you. All right. Thank you very much. You have made the point beautifully. Because for most, now interestingly, did anybody hear it differently? Did anybody hear this really differently? Yes. Others. We'll come back to that. That's really interesting. All right. So what happens for a typical native English speaker? We detect the interface. You were detecting it somewhere in here-ish, and that's about right for most typical human speakers. We would hear all of these notes here as ba, despite the fact that there's difference in timing, and we would hear all of these notes as pa, despite the fact that they're acoustically different. And the transition is pretty crisp. There's no in-between state. And thank you very much. Turns out that swamp sparrows do exactly the same thing in detecting the duration of their notes. So a study from 1989 went into the field. It's swamp sparrows, so you can imagine where they went. And the duration of notes was changed from 0 to 30, or back and forth in random increments. And moving from a short note of a 0 millisecond duration to a 10 millisecond duration, these birds expressed a behavioral response indicating that they were detecting no difference. And I'll be happy to tell you about that behavioral test. I've done it. It's fun to do. Field work is wonderful. And if you change note duration in the long note condition, then this variability is recognized as long note, and they have a transition, just like we just did, they have a categorical transition at a duration of around 13 milliseconds. So we know the behavioral answer. We know how the brain works at the perceptual level. Can we see how the brain works at the cellular level? So we went in and we took this syllable of the natural song of one note, two notes, very brief number 3, and number 4. So this is tweet, tweet, tweet. And we played it. We took this note and we replaced note number 3, which is naturally 7 milliseconds, and we replaced it with something that was either 4 milliseconds long, or 8 milliseconds long, or 16, or 27, or 31. The reason we chose these numbers is this is what those guys did in the field. We are doing exactly their study, except we're doing it in the laboratory. And just to advocate for these birds, according to birds, this song, that's a very distant bird apparently, sounds radically different than. So to a bird, those two things you just heard sound as distinct as ba and pa. So these guys are good, right? So we came along and we said, what would it look like if categorical perception were expressed on a cellular level? And we said, well, we know that this is a short note here, and this is a set of long notes here. So within category, you should respond the same way that you did to other elements of your category. In other words, the response to 4 and the response to 8 should be pretty similar to one another. And if we do the other thing, if we do the same thing for the other category, then the response to 16, 27, and 31 should look very similar to one another. Yet the response to blue should look very different than the response to red. That's what we expected to see. So we went in, we did this study, and we came close. We had a response to 4 and 8 that was very strong and very similar to one another, and a response to 27 and 31 that was very weak and very similar to one another. And then there's this guy. He's out of place. Because the response to 16 looks like it's telling us that 16 is a short note, but we know that 16 is a long note, according to that behavioral study performed in the wild. When we looked at a lot of cells and did this over and over, we found a neurophysiological category boundary at 21 milliseconds, whereas the behavioral boundary was at 13. This is either, depending on how you look at it, it's either unsatisfying, because it would suggest that the neurophysiology has nothing to do with perception, or it's challenging, in the sense that there may be something there that doesn't account for your observation. Can we figure out what that is? So we looked at the data and we saw that the behavioral study was done in upstate New York. Our birds were about 500 kilometers away in southwestern Pennsylvania. And we thought, you know, they're different birds. They're different communities, different populations, different breeding groups, etc. Is it the case that our birds are just different? So we went out into the field of southwestern Pennsylvania. This is what it looks like. We sneak into this is swamp sparrow heaven. They love this place. And we sneak in with a speaker, and we perform this behavioral test that enables them to reveal their categorical perception. And I like to say, if you can see the swamp sparrow in this picture, please see me regarding employment opportunities. So we do this study, and it turns out that when we did the study in Pennsylvania, when we took exactly the same methods as were used in New York, and we used them in Pennsylvania, we saw that the perceptual boundary in Pennsylvania was at 21 milliseconds. In New York, it's at 13. In Pennsylvania, it's at 21. And this, to me, was enormously satisfying. There's a field of study called neuroethology, in which you look at the neurobiology of animals doing what they do best. What are they naturalistically specialized to do, and how can we learn from that to understand our own condition? I think that's exactly what we did. We went in, we read a behavioral study from New York, we did a neurophysiological study in the lab, thought we got it wrong, actually went back out into the wild and learned something new about this population. And what we learned was that the boundary here is 13, the boundary there is 21, and it turns out we have come across regional dialects. They sound different, they recognize each other. The females from New York, they don't like Pennsylvania. The females from Pennsylvania, they don't like New York. And I actually got to talk to NPR about this, and I said that this is why Giants and Eagles fans don't get along. We finally figured it out. So we published this result, and this is, I'm always happy about this. Our local newspaper in Raleigh got wind of our results and teased us by publishing this political cartoon. So we had y'all come back and use guys and like totally chirp. So I'll cherish this for a long time. But we see then that these birds are learning, and not only are they learning, but they're learning exceedingly well. So now let's turn it back on ourselves. If your mom and dad are from Boston, they sound a certain way. And the reason they sound that way is that they've learned that Boston dialect, and probably so have you. If you're from the American South or from your favorite regional dialect site, you copy all the way down to those individual regional dialectical differences. And that's why we all sound the way that we do. Birds do the exact same thing. They are copying to an incredible scale. And when we look at the neurophysiology of this, we see some really interesting things start to emerge. In humans, we can have behavioral deficits that we call stuttering, and it's associated with deficits in the corticostriatal projections, among others. In songbirds, if I take that HVC into blue avian basal ganglia, a corticostriatal projection, and introduce an error in it, the birds repeat their sounds. Whether that's stuttering or not, we have to be careful. But it's awfully interesting that we could look into another system that has learned its vocalizations, which is actually exceedingly rare among animals. We do it, birds do it, parrots do it, hummingbirds do it, elephants, and a few others have come along. But your dog, for instance, does not. You can tell your dog sit, and he can learn to associate that command with a performance of an action. But he will never turn around and vocalize back to you, sit. So he can do sensory learning quite well, but he can't do that sensory motor learning. That's what we do exceedingly well, and now we have a model of another system that also does it exceedingly well, that we can study the neurobiology of that to perhaps understand ourselves better. So we have this, to summarize, then we have song-selective, temporally precise activity. The activity is related to the bird's perception, and it becomes a possible circuit basis of imitative learning. Now, let's switch gears, because we've been talking about these males. Let's go to the females, because they are not singing, but they are evaluating song quality to choose their mates, which brings up an interesting point. How can you decide whether something is good or bad if you yourself can't do it? That's an interesting question. So what we're going to do is we're going to look at these males, and we're going to have the male who is singing, and pink, a little heavy-handed, but the pink female is listening to this and trying to decide whether he's attractive or not. And long story made short, there's sort of an arms race going on here. Like the male can say, I am the best male, you so want to pick me. And it's her job to pierce through the manipulation to get to the truth. And so these are called honest signals. Honest signals in communication are what the female should be trying to find. Let's see if we can find one. So what could be making a song attractive? It could simply be that there are individual song features called sexy syllables. That there's something you do in your song, and you do it once, but you do it so well that it just pierces through and shines as a superlative song performance. That's true in some species, like canaries. It could be that it's complexity of song content. More is better. If you have more notes in your song or more song types in your repertoire, then you have a better neural system. You built a better neural system. You ate a lot of good food, or mom and dad fed you, so you come from good lineage, etc. Or it could be that it's a neuromuscular challenge. So for instance, right now I can clap very quickly with very small amplitude, or I can clap with enormous amplitude. But if I could clap very quickly with enormous amplitude, I can't fake that. I have to have superior musculature across my chest and shoulders. That would be an honest signal. I can tell you that I'm 8 feet tall and can beat up anybody in the American West. That's just talk. But if I can clap quickly, I have shown you an honest signal of my quality as a potential mate. So it turns out, when we look in the species where we do our neurophysiological recordings, it is this neuromuscular challenge that matters. It is some combination of broad frequency bandwidth, sweeping from a high note down to a low note, or from a low note up to a high note, combined with rapid tempo. It's not altogether different than a big physical excursion done quickly. So then we started asking, what matters more? If it's some combination of frequency and tempo, which one matters? So to do that, we manipulated them. And we have songs that hopefully we'll be able to hear this distant bird a little better. Here's the natural song. This bird is underwater, apparently. All right. So now what we're going to do is we're going to take this and then we're going to speed it up. So here's the original. Faster. And slower. So we can manipulate tempo but not frequency with fancy software. Here's the original song. We'll change its pitch, its frequency, and we'll do it again. So So we can take each of these parameters and separate them out and ask, okay, females, if it's some combination of frequency and tempo, when we manipulate one, which one matters more? And I was certain, as scientists often are, that it was going to be a pipe organ. Yeah, sorry, a little commentary there. It was going to be basically a pipe organ analogy, in the sense that this pipe organ has the ability to produce different notes because it has different chambers of different sizes with different resonance properties. If you have a lower frequency song, you would have bigger pipes, you would be a physically larger male, you would likely be more likely to win any territorial encounter, you could probably defend territory better, maybe provide food better, etc. So I was certain that frequency was going to be the thing that answered this question, and I was dead wrong. So we plot here on the y-axis the female's preference in a behavioral test. Here she's preferring the altered song if she goes up, and here she's preferring the natural song if she goes down, and here we're changing the note frequency either down 200 cents, which would be do, re, mi, or re, two notes down in our nomenclature, and she has very little relation, no significant relation between changing frequency and changing her preference. Swing and a miss. So what about the other one? What about tempo? And it turns out we hit a really interesting result. That now, when we change tempo, we're changing the tempo here. We've slowed it down from 100% to 50%, and here we're changing it from 100% to 150 to 200%. So we're going faster over here and slower over here. And here she prefers altered, here she prefers natural, and we get a result that is largely like that. So as you go faster and faster, the female prefers it more and more. We're keeping the same excursion, but we're just doing it artificially rapidly. And she's able to detect that and expresses a significantly greater preference for that modulated song. But what, to my mind, was an awfully interesting result is that not all females agree. So some say that a faster song is in fact less attractive, which raises interesting implications for the evolution of this song. That if you have all females agree that taller is better, then the human population will tend towards getting taller because of the selection. But if some females like tall, some females like short, some females like brunette, blonde, etc. If there's diversity in preferences, then diversity in quality will persist. So it's not a singular answer of faster is better. It has to do, I can't tell you this whole story, but I'll tell you the punchline. It has to do with the female's experience when she was young, what she learned to value. So she's getting it individually right, according to her standards, but not all females agree. So then we said, all right, what can we learn about the neurophysiology of this? This is really interesting. Let's take our little device and let's go back in and record this neurophysiology. And here we're playing the song, and the song is occurring here, and it's quiet here and quiet here. And I'm recording this neuron, and it's popping along. And if I summarize its activity across a lot of trials, then during the quiet, then the neuron is marginally active, if at all. Then I turn on the song, and the response gets much greater, and I turn the song off, and the response goes back down. This is one cell from one bird. But we can summarize this as the strength of this response. In other words, how big is this plateau? And it turns out that it's greatest for the female's most preferred song. When we play her a buffet of options, we can define her least preferred song. We can have song of other species, which it would behoove her not to mate with other species. And then we can have white noise, which is just a psh sound. Now, interestingly, remember, not all females agree. So what this most preferred song is varies across females. But this neurophysiological relation persists. So we have found an area of the brain that is not simply responding to the acoustics of the signal, but is responding to the evaluation, the subjective value of the quality of a sensory experience. To my mind, that's awfully interesting. So now we're starting to ask, the grant that we have is broadly hinging on how does perception of value get linked to initiation of action so that you choose one possible outcome from among many possible alternatives. It's one thing for me to persuade you that a Big Mac smells good. It's another thing entirely for me to persuade you to open your wallet and give me money. So how is it that that link occurs? And the neurons that I've been telling you about are in a place called CM, and they're literally at the hub of this diagram. But we found an interrelation between CM and other places that could possibly contribute to this. We have other areas that are associated with perception and evaluation in blue. We have this place called UVA, which is auditory visual integration, which means now that she hears a song and she likes it, and she saw that guy perform it, so now she knows that's my mate. Auditory visual integration could be performed possibly in contribution by UVA. The hypothalamus is a hormonal master control center and can indicate postural indicators of preference. And by this, I mean that song is so important to a female bird that she will solicit copulation from a speaker, even if no male is physically present. So there's a postural, when I say solicit copulation, I mean a postural indicator of mate choice. Then she will also, in a study we published, she will call more times in response to the song she liked best. So there are vocal indicators of preference. And then there's the structure called the ventral tegmental area, the VTA, which is massively interesting from a human perspective because this has to do with feelings of value, feelings of reward, feelings of pleasure. This is a reward-related pathway. And we know that there are connections from CM to the reward center, connections from CM indirectly to the vocal center, over to the postural indicator center. It doesn't mean that there's a smoking gun and we found it, thank you, good night, but it does mean that we have found a context in which we should set up shop to look for mechanistic means through which perception drives action. And that's what we're doing. So just in the last few minutes here, I want to give a bit of a praise to basic science in general, because a lot of medicine, a lot of biomedical science now is driven exclusively towards medicine. And I think that this is a beautiful example of how basic science can, in fact, transform medicine in a really beneficial way. Because this is green algae, and in the 1990s, researchers were looking at green algae and saying, how is it that green algae are responsive to light? Can we understand that? And it turns out that they did. And green algae this is a cell now. This is the outside of the cell, and this is the inside of the cell. So here is the cell membrane. And green algae have on them these little proteins which act like straws. And normally the straw is clenched shut, and nothing can get in or out. But when light shines on this one, then plus charges can go inside and turn the cell on. Interestingly, this type of transmembrane channel, when you shine blue light on it, you let positives in and you can turn a cell on. Another type of transmembrane channel, when you shine green light on it, it opens up and lets pluses out and turns the cell off. So these people who were studying algae published the results, and some very clever neurobiologists looked at this and said, this is gigantic. This is an opportunity to use light to turn cells on and off. Because the challenge in neurobiology is to say that a certain area is linked to a certain aspect of behavior. That's a really challenging thing to do. But if you have the ability to go in and experimentally manipulate one population of cells and only that population of cells during a behavioral test, then you can begin to gain insight into what that set of cells does. The neurobiologists who looked at this algae literature, why they were perusing the algae literature, I don't know, but thank goodness they were. They took this into neurobiology and have used enemies of humans, like viruses, to induce neurons to begin expressing these transmembrane channels. And all of a sudden, we have the ability to use light to turn cells on and off. Here is an example of turning cells on in 4 different ways. You can flash the light of pop, 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 and it fires this way. You flash the light a little faster, it goes faster. You flash the light faster, it goes faster, etc. And it's tough to see, but there are spikes going on here. And you turn on the green light, there are no spikes there. And then you turn the green light off and spikes come back. So we now have the ability to use light to jack into the activity of a very specific set of neurons and begin asking about their functional consequences. And with what we have in terms of this physical stimulus evaluation pathway, we are awfully curious to know how that translates into the initiation of action, or not the initiation of action. So in summary, what we have is we've defined the features that make song attractive. We have circuitry linking the perception to action. We have patterns through which perception and action may be encoded. And broadly speaking, this is a model to understand the mechanisms through which sensory experience directs motor activation. And if we step back and think more broadly about it, this is a model of how we make decisions. Because sensory-evoked activation or sensory-evoked failure to act is a very important part of our lives. It keeps us safe, keeps us healthy, keeps us wealthy, we hope. And then if we can understand possibly the mechanisms of how the system is right, then we could possibly understand mechanisms of how the system goes wrong. So for instance, addictive relapse is a huge problem. That an addict will have stimuli that are associated with their previous addiction experience. That house, or that smell, or my buddy always wore that green pullover when we did drugs together, or whatever. Mercifully, I don't have any addiction experience, but there's a moment of intense craving. And if you can get an addict past that moment, then it will pass. And so the dream, when I allow myself to dream such things, is to understand the mechanisms of this pathway and gain insight into perhaps a brief pharmacological manipulation to get you past that moment of intense craving. That would have wonderfully beneficial consequences for our society. So I put this up here as thank you to a lot of people, and on the left especially, all of these people on the left except for the two at the top. The two at the top are grad students. All of those on the left are undergraduates. Those that are in italics have worked so hard in our lab that they have become authors on scientific manuscripts. To my mind, as an undergrad, that's awesome. Those that are underlined have done not only that, but they've won grants in support of their own research. So these are sharp kids, and they deserve to be publicly praised, so there you go. So I'll be happy to take questions. Yes, sir.
Delineated the idea that there's learned behavior and potentially either same cells or differentiated cells that loop back in that thing. And of course, then there's cells that are, I guess, purely instinctual. Have you worked on the idea that raising a chick with a different species where you would learn a different song would then find itself attracted to or responsive to that other species rather than its native species?
You have just made me so happy. Did everyone hear the question? Oh, so in distillation, if you took a chick that was genetically a sparrow and raised it environmentally as a finch, what would it prefer? Is that a fair? And it turns out two answers to that. One is that I know the answer, and I can give you a sobering answer. Is that if you let's take a male before we do a female, and you can take a male who is genetically X and raise him acoustically Y, and he will be acoustically Y, unless you leave his cage door open, which I've done. And when he hears his own species, he somehow knows, and he goes home. So if you raise him as Y and he ever hears X, he goes home to X and stays put. If you raise him as X and then try to persuade him to become Y, he will never leave X. So it's not a completely flat playing surface, right? And the females, it turns out that I have a colleague now in Florida, and she and I asked exactly that same question when we were sitting in the swamps of Pennsylvania, and she went out and did that study, and it turns out she collected birds from the wild, and when they were very young, as genetically this population, and she played them acoustically some other population. They prefer their genetic population. So again, it's not an entirely flat playing surface, which is awfully interesting to think about. I can't answer it for you yet, but thank you, a good question. Yeah.
So do you know why the female baby birds do not learn the songs that they hear from the adults?
There's a group of birds called songbirds, and those are capable of learning. Then there are others which are non-songbirds. They're not capable of learning. But within those that are capable of learning their songs, when they hatch, males and females begin to build the associated brain structures. And then very early on in development, the females have those brain structures atrophy, and the males go on to build a very complex system that I showed you in that map. If you perform a hormonal manipulation of a genetic female such that you give her the induction of that system, she will then go on to sing. And interestingly, she will not sing what she prefers. That's awfully interesting also. Good. Yeah.
Do the birds have a significant hearing? Are they really good at hearing just within a certain range? And the reason I'm asking is, how can a cat with a bell on it catch a bird if they can hear such details that you're explaining here?
We share features in that if we plot frequency with a very low frequency here and a very high frequency here, and then we plot the amount of intensity of sound that is necessary to turn on your hearing cells, then at very low frequencies, below about 20 hertz, it takes more energy than your system can take. In other words, you can't really hear below 20 hertz. And over some odd number of 13,000 to 20,000, you can't hear above that. But below that is a very sensitive region. And there are two interesting aspects of that. One, it's awfully neat that the songs use their power and their energy and transmit within that especially sensitive range. So we do that also. We transmit energy in the range to which we are acoustically most sensitive. That's just convenient. And two, there's this little device called a mosquito. Has anybody heard about this device called a mosquito? So it turns out that humans, we typically, when we're very young, we can hear up to about 20,000 hertz. But as you get older and older and older, you can only hear down to maybe 11,000 hertz or something like that. So there's a device called a mosquito, which is used by some vendors that will transmit at about 17,000 hertz, and it bugs teenagers. But the old people who have money to spend money in your store don't get bugged. It's wonderful. Somebody made a lot of money off that idea.
You were talking about the differences in the females' preferences for the songs they learn. Did you say that they grow up preferring adult males who are like their fathers?
All right, I got to be careful because this could get strange quickly. In our literature, there's this term preference, and it has sort of polluted our literature because let's imagine a young human female that she, all we do is we observe where she goes. She might sit close to her father. She might sit close to her brother. She might sit close to her boyfriend. But her feelings, beyond simple affiliation, her feelings towards each of those individuals are quite different. And so we are careful in our studies to look at mate choice as made evident by solicitation of copulation. So there you go. Now, when she said, if I did say that she prefers the song of her father, I slipped and used a polluted literature term. She likes her father, we presume. What's going on inside, we have to be careful. We don't actually know what it's like to be a bird. But she will express affiliative behaviors in association with her father. And then with regard to what she learns when she's little, there was a really interesting study done once, hasn't been replicated, but let's imagine a human example that I'm going to say Brad Pitt is universally attractive. Is that true? Would you guys grant me that? So if you were a woman and you encounter Brad Pitt, let's say that you perceive yourself as a 7 and he's a 10. So you wouldn't fool with him because he's out of your league. You probably couldn't get him, and if you did, you couldn't keep him. So it's not really that efficient a move to go reaching for that 10. And so what you're supposedly better served to do is to take just above your station. So people have looked at birds, and they've raised them in an environment where the clutch of eggs was very large, 8 eggs, and mom and dad are busy. They can only feed those 8 so much, and so you end up being what is called a low-quality offspring. Then in another clutch of eggs, there were only 2 or 3. So mom and dad now have much more time and energy and food to devote to you, and the result is a high-quality offspring. So we then have high-quality males and high-quality females and low-quality males and low-quality females. And in this study, when low-quality females were provided a high-quality male or a low-quality male, they almost always chose the low-quality male.
You're talking that birds have the equivalent of self-esteem?
Well, wait, this is where, that's why I say it's only been one study, but they took a high-quality female and they offered her a high-quality male or a low-quality female, and she chose the high-quality male. Then, in this study, they took a high-quality female, trimmed her feathers 1/2 centimeter, and let her fly around and figure out that she was altered, and she then began preferring a low-quality male. It's only one, but it's an awfully interesting result. All right. Well, thank you guys. Thanks very much.